Good morning, everyone. I'm Christopher Tompkins, Chromatid's Chief Technology Officer. With me today is Aaron Cross, our VP of Research and Development. We would both like to welcome you to Chromatid's inaugural webinar, where today we are taking a deep dive into one of the whole genome single-cell techniques from our directional genomic hybridization platform, dGH Screen. Now, dGH Screen is available today as a research service and soon will be available in kit form for use in your laboratories. In this cartoon view of a single dGH screen metaphase chromosome, the bare chromosome is shown in blue and the pink represents the fluorescent signal of millions of dGH probes, which we hybridize or paint on chromosomes to map out the underlying DNA. But before we take too deep of a dive into dGH Screen, Chromatid does offer a comprehensive portfolio for mapping structure and structural variation in genomes. And I thought I would just briefly touch on the relationships between all of our various products and services. Besides dGH Screen, we offer dGH Discover and dGH Insight, which differ only in the fluorescence labeling strategy and the amount of the genome covered in a single assay. Screen, for instance, uses millions of unique probes to detect structural variation anywhere in the genome. dGH Discover uses hundreds or thousands of probes to cover individual chromosomes with banding patterns, allowing us to both visualize structural variants and to locate the breakpoints of inversions and translocations. dGH Insight then uses thousands of hybridization probes to target localized regions within the genome, for instance, oncogenes, edit sites, or lentiviral inserts. To round out our metaphase techniques, we offer G-banding services. G-banding is a low-resolution staining technique that is valuable because it's both widely accepted and also because it's orthogonal to any genomic method, including dGH and NGS sequencing. As metaphase methods, all of the dGH tools require live and dividing cells. If your samples won't divide or are preserved, we offer a ultra-high-resolution FISH technique called pinpoint FISH, which uses the dGH Insight probes in interphase cells. We perform pinpoint FISH as a service, or you can buy pinpoint FISH kits directly from us or through our growing distributor network today. And finally, we've just launched custom plasmid manufacturing services, so we can support our cell and gene therapy customers by both providing vectors and measuring the genomic structural effects of those vectors on edited genomes. Now, if we move away from the cartoons for a moment, the workflow for dGH parallels any metaphase FISH assay. A metaphase spread is dropped on a microscope slide, imaged, and then the chromosomes from each individual cell are arranged in silico into a karyogram view. The data from many cells are then summarized to give not just an average prevalence of structural variation across the batch of cells, but instead a cell-by-cell, chromosome-by-chromosome distribution of structural variation such as translocations and inversions. For genotox studies in cell and gene therapy, for instance, we would typically provide edit site translocations, random structural variations, and other measures of genomic instability. Hit pause, William.